KarMMa-3 trial was an important clinical study because it compared the efficacy of the current standards of care, so that means drug-based regimens, with CAR-T product called ide-cel in patients with relapsed and refractory multiple myeloma. So that study enrolled nearly 400 patients who had received 2 to 4 prior lines of treatment. And all those patients were triple class exposed. That means they were exposed to a proteasome inhibitor, an immunomodulatory drug, and an anti-CD38 monoclonal antibody, which was daratumumab. So those patients were heavily pretreated because in the KarMMa study, patients had received a median of 3 prior lines of therapy. Around 2/3 of the patients were already triple class refractory. And that study looked at the efficacy of continuous drug-based treatment, and patients could receive 1 out of 5 different possible treatments compared to only 1 single infusion of ide-cel. So the primary endpoint, as it is called, of the KarMMa-3 study was to measure is there any difference in progression-free survival or PFS. And then there were also a lot of secondary endpoints, including the overall survival, but also the tolerability. So in other words, potential side effects. The primary endpoint, so the PFS, showed a clear and statistically significant benefit in favor of 1 infusion of ide-cel compared to a continuous drug-based treatment. And this was not only highly statistically significant, but also highly clinically relevant for patients. Of course, the CAR-T cell infusion is associated with specific side effects like cytokine release syndrome, CRS, or neurotoxicity called ICANS. But one could say that the risk for getting severe CRS or severe ICANS was extremely low in the ide-cel arm. In that study, one of the secondary endpoints was also measurement of health-related quality of life. And that's very important because irrespective of high clinical efficacy, you also want to evaluate how do patients experience this particular type of treatment. And so patient-reported outcome measurement is therefore very important. And we did that through standard evaluation methods for quality of life measurements. And these are measurements derived from the EORTC. So you have the EORTC QLQ-C30 module, which measures 15 different domains in that study. We had also a specific module for myeloma, which is called the QLQ Myeloma 20 model, which encompasses 4 domains. And there is also a kind of a health utility index, which is called the EQ5D5L. So we used those instruments to measure the quality of life in both study arms, in the arm with ide-cel and the control arm with standard of care treatment. What we do know from the historical data is that with the currently available treatment options for patients with relapsed refractory multiple myeloma, the best one can achieve is a kind of stabilization of quality of life. And in this study, it's of particular importance to see if 1 single infusion of a CAR-T cell product compared to a continuous treatment with drugs has an important impact on the quality of life of patients. For instance, we show that patients who received ide-cel had a rapid improvement in their global health status. They had much more reduction in disease-related symptoms like fatigue and pain compared to the control arm. They had improvement in physical functioning and cognitive, so mental functioning, compared to patients in the control arm. And that's so important because many patients tell us that the time after the CAR-T infusion, so when they are off treatment, is the best quality time during their whole disease journey. So coming back to what this will bring for the future, I think it shows us the power of CAR-T treatment, not only in terms of clinical efficacy, but also in terms of impact on quality of life for patients. And in addition, it also shows us the value of measurement of patient-reported outcomes by health-related quality of life in all the types of clinical studies we are doing.